Gene regulation essentially refers to our ability to switch off genes and switch on genes depending on when we need to switch them on and off. It's a way of energy conservation because there are going to be times in your life cycle where you don't need the creation of certain proteins. So it would be a waste of energy and resources within the body to create proteins that you are not essentially needing. So gene regulation enables the turning on of genes and turning off of genes depending on when we need them or not. So this process of regulation starts very, very shortly after we've become like an egg cell. When the gene regulation starts to take hold of our cell and become more diverse with different functions as they form our body. This process is called morphogenesis. So, where you've got your egg cell and all of the you've got your egg cell which starts to divide into cells using the mitosis process. Each of these cells then needs to become specialized. So some of these cells will then grow on to become brain cells, liver cells, stem cells, muscle cells, whatever you need. That process of cell specialization is all controlled by gene regulation. So, mismanagement of regulation is essentially what causes cancers or other terrible pathologies or conditions and things like that. If you think about two separate populations of cells, so for example, in this little diagram here, we've got a stomach cell and it's got a stomach cell ending up with hydrochloric acid. So hydrochloric acid is one of our stomach acids that helps digest food. So this eye cell has just created a protein that helps with the production of hydrochloric acid. So the eye cell department is completely producing hydrochloric acid when this is supposed to be for the stomach cell. The eye cell gets confused because it doesn't think that it's on the stomach and ends up burning the eyes of the human. So gene regulation is really important so it doesn't create proteins that don't need to be in locations that they don't need to be in. Um, so for example, we've got stomach epithelial cells that create gastric acid, so like the ones in our little picture there, and cone cells in the retina of our eyes that are responsible for us being able to see color. If for some reason the regulation of genes in the cone cell failed, it would become the cancer of the eye, or uncontrolled protein creation in the eye. Another possibility would be that the cone cell starts to downregulate its normal cone cell genes, so the genes that are able to see color, and upregulate or stimulate the genes that are found in stomach cells for some reason. And therefore, the cone cell will start to transform into a stomach cell, doing all of the normal jobs that an epithelial cell would do, secreting gastric acid and absorbing nutrients. And this is a huge problem because obviously we don't want anything that the stomach does to be happening in our eyes. So I mentioned two words previously, one of them called upregulation and the other one called downregulation. These are both determined by something called the activator and the repressor. So next, these activation and repressor functions are carried out during the transcription stage of protein synthesis. So transcription actually ends up being a pretty key regulatory point for all of our genes. There are things called transcription factor proteins. So these are proteins that specifically stimulate the transcription of a gene or downregulate or inhibit the transcription of a gene. They will bind to specific DNA sequences in or near a gene and either stimulate or repress the transcription into RNA. Basically, they bind to parts of DNA that stop specific genes from being transcribed or increase the rate that we're being transcribed. So if I get the laser pointer back up, we've got this pink protein binding onto a site. This is a repressor protein, right? So it actually stops all of the mRNA from being accessed and it doesn't result in the transcription of the target gene. The repressor blocks the general transcription factors and RNA polymerase from actually hooking itself onto the DNA strand. So no transcription can occur and this protein will not be expressed. On the other side, we've got activators. So these are like our green sites here. it will activate onto a binding site that hooks RNA polymerase onto the DNA strand itself, which lets the RNA polymerase transcribe the target gene, which is in purple over here. As these transcription factors are also proteins encoded by the DNA, they can actually have transcription factors that regulate them as well. So it becomes this like pathway of proteins influencing genes. However, that's a little bit more complicated than you guys need to know. The genes that encode for a transcription factor are called regulatory genes, as they are encoding for these regulatory factors which control the expression of functional genes. When we talked about growth factors before, um, and sorry, when we talked before about growth factors, these actually act via a very similar mechanism, where their signaling pathways cause gene expression to be promoted or inhibited by signaling the action of activator or repressor proteins. So there are a couple of different kinds of gene regulation that QCAA expects you to know. The first one is about how sex is determined in humans using something called the SRY gene or the sex gene. Most of your chromosomes come in homologous pairs. So these are identical pairs that we talked about in the mitosis, meiosis, cellular replication step. The two chromosomes of a homologous pair contain the same basic information. That is, the same pair of genes in the same order organized across the chromosome. But many carry different versions of those genes. So if any of you guys have done grade 10, grade 10 biology, you would know about dominant and recessive genes. They can carry these chromosomes can carry the same kind of gene, but one chromosome may carry the dominant version of the gene or the dominant allele, and the second version of the gene may carry the recessive allele. So there is only one set of chromosomes that isn't arranged in a homologous pair, and that is your sex chromosomes. So for people who are XX, so typically with female reproductive organs, they have the opportunity to have homologous chromosomes because they're the same size, so each of these genes will be organized in the same way. Males, on the other hand, with XY chromosomes, will have different sized sex chromosomes because the Y chromosome is a lot smaller than the X. And they aren't considered homologous. So instead of an X and a Y, a human female will have two X chromosomes. Because X chromosomes do actually form a homologous pair, these genes can actually because X chromosomes do not always come in homologous pairs, these genes they carry show unique and distinctive patterns of inheritance. So again, if you guys remember doing things with pedigree charts, you would have known that there would be like sex-linked genes, where the chance of you getting this particular mutation would be higher depending on whether you had whether you were a male or a female. So human X and Y chromosomes determine the biological sex of a person, with XX specifying female and XY specifying male. Although the Y chromosome contains a small region of similarity to the X chromosome, here you can see that both of them have at least one of the X chromosomes, and the Y contains some similarity to the X chromosome. The Y chromosome is much shorter and contains way fewer genes. To put some numbers to it, the X chromosome has about 800 to 900 protein coding genes with a really, really wide variety of functions, while the Y chromosome only has 60 to 70 protein coding genes, about half of which are active only in sperm producing organs or in the testicles. The human Y chromosome plays a key role in determining the sex of a developing embryo. This is mostly due to a gene called SRY, or the sex determining region of the Y gene. SRY is found on the Y chromosome and encodes a protein that turns on the other genes required for male development. XX embryos typically do not have an SRY, so this is our typical female chromosome orientation there. They do not have the SRY gene, so they will develop as female, and all of the genes that stimulate female reproductive organs and female sex characteristics will start to be switched on. If an SRY bearing X chromosome fertilizes a normal egg, it will produce an atypical or chromosomally an atypical female embryo. So it'll still have an XX chromosome combination, but because it's got the SRY gene, this individual will start forming male sex characteristics. Um, so this is a instance of intersex humans developing, basically. If an SRY deficient Y chromosome, so this is our XY, um, this is an individual with an XY combination, but it doesn't have the SRI gene. It won't start producing male characteristics. Instead, it'll be a female with XY chromosomes. Another an example of intersex. So if you want to think about it simply, the default sex characteristic will always be female sex characteristics because most of the mass that you're getting from the zygote is from the female egg cell. Male sex characteristics will only start to develop if the SRI gene is present. And if it's present in where it shouldn't be present, male with male sex characteristics will only have an XX chromosome combination, and a female without the SRI gene will have an XY chromosome combination. So there, in terms of what the SRY gene is actually doing, it encodes a protein called the SRY protein, which is a regulatory protein. It acts as a transcription factor, which causes the upregulation of other transcription factors. One important transcription factor that it upregulates is the SOX9. Sorry, just give me a second. SOX9, which is responsible for turning a population of undifferentiated cells into specialized cells, which go on to forming the testes in males. It also helps with the development and maturation of the testes and male reproductive organs, which eventually go on to secrete testosterone and other male hormones, which eventually develop male sex characteristics. If this gene is not present, so if there is no SRY gene, you end up with no production of testosterone and no production of male sex characteristics. So by default, even if they've got XY chromosomes, the human that develops will have female sex characteristics and female anatomy. So the next type of transcription factor that the QCAA wants you to have a look at is called Hox genes or homeobox genes. These genes are controlling the structures of the body or like sections of the physical body. And when misexpressed, it can cause some really, really crazy mutations. Now, the main model of Hox genes, when they're usually studied, are a fruit fly. And typically, a fruit fly will have 6 legs. These legs usually grow out of the thorax, as you can see labeled B. So all 3 of these appendages are coming out of this midsection here, the thorax. And it is the Hox gene that is responsible for the development of these structures in the correct location. There are other regulatory genes that are involved, but they're all sort of controlled by the Hox gene. The genes that are determining the identity of particular segments, as in what ends up growing as the head, what ends up growing as the thorax, and what ends up growing as the abdomen, are all all dictate what cells go into which region of the body. So if you consider a particular type of Hox gene, this Hox gene in the fly, it is responsible for the formation of the thorax of the insect. The gene here acts as a master regulator and activates all of the genes responsible for creating this fly's second pair of legs and the other structures specific to the second thorax segment. If the gene is not correctly expressed here, we get a normal, if the gene is correctly expressed here, we get a normal looking fly with 3 sets of legs coming out of the thorax. However, if this gene is expressed in the wrong section, which, happens after a mutation, some pretty strange things can happen. Say, for example, this fly had a mutation and it caused the Hox gene to grow in the fly's head or be expressed in the fly's head instead of the antenna. The gene activating the program would typically develop the second segment of the thorax and its associated features in the wrong place entirely. So what you essentially see there is the antenna of the fly antenna of the fly growing out of the head, growing as legs instead. So mutations in the Hox gene cause abnormal structures to form in different segments of the body, basically.